welcome everyone to the AlphaFold webinar series. So last year we saw some spectacular developments in the field of protein structure prediction with both AlphaFold and RosettaFold published almost a year ago now on 15th of July. And this was followed by the launch of a new resource, AlphaFold Database, to make now almost 1 million structures available, and soon it might be more than hundreds of million. And these are all high-quality predicted protein structures. So the impact has been enormous, and if we just look at the number of publications, that's astonishing. So in less than a year, we have seen 700 published papers, 150 preprints, and astonishingly 300 reviews that mention AlphaFold. And this is less than a year after all this became available. And so we thought this webinar series would be a nice way of to mark these amazing developments. And over these 3 webinars, we are going to hear about the impact these developments have had on obviously the research in life science. But I also want to highlight the second webinar next week, where we want to look at how this impacts teaching and training going forward, and what new things we need to develop, or what material is already available in terms of teaching and training. And the last webinar then will look at very specific use cases for research and development in life sciences. And then there will be a panel discussion. So if you haven't registered for those, please register. And let's start with this webinar, which will give us a bit more information on the scope and vision of AlphaFold. And I'm really happy to introduce Kathryn, who works in the AlphaFold team at DeepMind. And today she is going to give an overview of AlphaFold system and review the ways in which the system has been built upon by others, and also discuss how to evaluate AlphaFold for a new application. Over to you, Kathryn.
Thanks, Samir. Great. So can you just sort of nod and confirm that the slide sharing is going? Okay, great. Cool. So as Samir said, I'm Kathryn Tunyasuvunakool. I'm a research scientist at DeepMind, and I've done work on, for instance, the application of AlphaFold to the human proteome, and also I was involved in the AlphaFold Protein Structure Database work that Samir mentioned. So first, I'd like to thank EMBL-EBI for organizing another of these webinars and for inviting me to speak today. The plan is for me to start by providing sort of the general background on AlphaFold and its predictions. So for anyone less familiar with the method, that will set you up for hearing from the other speakers in this series. Then in the second half, I'll talk a bit about a new piece of DeepMind work that's been published since the last webinar, which builds on AlphaFold to predict the structure of protein complexes. And finally, I'll give some updates on accessing predictions, including things that are new across our database, the Colab, and the open source code. So starting with the overview then. AlphaFold was developed by DeepMind, which is an AI research company based here in London. And it's always been one of DeepMind's goals to apply AI to make a positive impact in the real world. So for example, by accelerating scientific research. And so a team at the company chose to work on this one specific scientific challenge of predicting a protein's 3D structure from its amino acid sequence. And AlphaFold is the resulting ML model that aims to solve that problem. It became widely known after the 14th CASP, which is an event that runs every 2 years in this field to assess progress. And basically, CASP entrants have to predict a set of about 100 protein structures, which have been solved experimentally, but have yet to appear in PDB. And on the right, you can see a couple of AlphaFold's CASP14 predictions, which got people very interested. Our prediction in the blue, and then the experimentally determined structure in green. So the model itself takes 3 inputs. First, the target amino acid sequence, then a multiple sequence alignment, or MSA, containing sequences evolutionarily related to the target. And finally, we have a small number of known protein structures for use as templates. For constructing the MSAs and for finding templates, we can use fairly standard tools and databases. And it's worth noting that AlphaFold isn't reliant on templates. The model can sometimes generate good predictions without them if it has an informative MSA. And it can also ignore templates if it thinks that they aren't useful. It isn't forced to copy their structures. The core of the model is a network that we call evoformer, which is repeated 48 times. And the goal of this part of the network is to progressively update a mathematical representation of the MSA and a representation of all residue pairs, allowing each one to inform the other. And then over the course of all these layers, that builds up an informative picture of how residues are related to one another. Next, the structure module turns that information into an actual predicted structure. And based on the final representations, that is predicting a rotation and a translation per residue in order to place each one individually within 3D space. A small network on top of that predicts the chi angles so that we can generate side chains. And then finally, the predicted structure is relaxed, which means that we run it through an energy minimization process. The point of that is to fix any remaining stereochemical violations, things like unrealistic bond lengths or angles, which are usually pretty minor within the confident parts of the structure. One interesting thing that we found when developing AlphaFold was that feeding certain outputs of the network back through again as inputs improved accuracy. We term that recycling, which turns the structure prediction process into something iterative. And by default, we run 3 of these recycling iterations. So it's worth noting that there are a couple of places here where a user could intervene. So for example, you could imagine making modifications to the input MSA, or providing carefully selected templates, or altering the number of recyclings. And those are all modifications that the community has experimented with since launch. Finally, for this overview, AlphaFold doesn't just output a structure prediction, it also produces 2 metrics to help you judge the model's confidence. So we have our local confidence metric, PLDDT, and we have a pairwise confidence metric, PAE. Those metrics are really key for interpreting predictions correctly, and so they'll be the focus of this next section. Starting with PLDDT, this is AlphaFold's per residue prediction of its LDDT CA score. Now, LDDT was a pre-existing metric in structure prediction, and the main thing to realize about it is that it doesn't measure how well the entire predicted and true structures can be aligned on top of each other. Instead, LDDT rewards predictions that are locally correct, so things that get the individual domains correct, even if their relative placement is off. And PLDDT behaves very similarly. So PLDDT runs on a scale from 0 to 100, where 100 is the most confident. And across all of our resources, we color predictions based on these 4 confidence bands. So for the prediction on the left, we can conclude that we have a confidently predicted domain in the middle there, with PLDDT above 70 almost everywhere. We also have this long tail with very low PLDDT. And our advice is that you shouldn't really treat this as a structure prediction at all, but rather as a disorder prediction. There's evidence to suggest that some of these very low PLDDT regions are in fact unstructured in isolation. The example on the right illustrates the biggest mistake we see with PLDDT. Here we have a protein that looks like beads on a string. So it's like a chain of domains that may be connected by flexible linkers. And just because all of these domains individually have high PLDDT, that doesn't mean that we know anything about their relative positions. In order to assess inter-domain confidence, you actually need to use a different metric. And that's why we also provide this predicted aligned error, or PAE. So PAE was developed specifically to help judge AlphaFold's confidence in relative domain positions. And in short, it's a pairwise metric. So it will give you 1 number per pair of residues X and Y. If you were to imagine aligning the prediction and the true structure on residue X, this is going to tell you the expected error in the position of residue Y over here. And we'd normally present this on a 2D plot like so, with residue number running along both axes. And the darker green here corresponds to higher AlphaFold confidence. So for the little 2-domain protein that's shown, the high confidence green squares correspond to the individual domains. because for any two residues within the same domain, we're pretty confident in their relative position. However, if you look at a pair of residues from different domains, you can see that we don't have high confidence in their relative position. And that means that for this structure, the domain positions are essentially almost random and they shouldn't be interpreted. Looking at another couple of examples really quickly, on the left, we have that beads on a string type protein from earlier. And you see that if you take a pair of residues X and Y from different domains and look at that location on the PAE plot, there's no dark green color. So AlphaFold doesn't have any opinion about their relative positions. Whereas on the right is an example of a potential domain packing prediction. And here, if we take a pair of residues from different domains that seem to be close, and we look those up on the PAE plot, we do see a dark green patch. So that indicates that AlphaFold really does predict that those two residues are close or in contact. It's not just that the network had to put those domains somewhere and they happen to end up close to each other. One final point, not so much about interpreting individual predictions, but about how to evaluate AlphaFold's performance in general. I often get asked, how does AlphaFold perform for a particular task? And the best way to find out is usually to put together a suitable test set of proteins, just run AlphaFold over those and measure the accuracy yourself. But there are some basic points to bear in mind when you're setting up an evaluation like that. The main one is that ML models are expected to perform well on any input that they've encountered during training. For those examples, the model has basically seen the correct answer, and it can essentially memorize it and parrot it back without needing to learn anything fundamental. And so AlphaFold was trained on PDB structures released before the 30th of April, 2018. So any structure released before that date shouldn't really be used for evaluation. It's too easy. Secondly, test cases that are very similar to something in training also tend to be a bit too easy and less informative. So in biology, that's a particular issue because we have evolution and we have homology. So to put together a challenging test set, it may be worth filtering out the proteins that are too similar to pre-2018 PDB entries, based on something like identity and coverage of the best template. And finally, if you're using templates to make your predictions, it's actually quite easy to accidentally pass in the true structure from PDB as a template. And to avoid this, we do provide a max template date flag, which you should set to restrict the templates that are available. I actually originally planned to say much more about model evaluation, something I'm very interested in, but then I realized I didn't quite have time. So if anyone was hoping to hear more about that, just ask in the Q&A. Okay, so now that we're done reviewing the basics, I'm very excited to talk about some DeepMind work made public since our last webinar. And that's AlphaFold-Multimer, a new model for protein complex prediction. The rationale for working on complexes is really quite clear. Most interesting biology isn't coming from one protein, but rather from complex systems of many components. And so if we can extend our method from single chains to complexes, then that opens the door to investigating much more biologically relevant systems, like, say, large molecular machines or interaction networks. And in fact, complexes are so important that within about a week of the original AlphaFold launch, I think it was, the community had already come up with some clever hacks for generating complex predictions. So for instance, just joining all of the sequences together with linkers, then running the lot through AlphaFold. And while that does work surprisingly well, we think we've managed to substantially improve on it in AlphaFold-Multimer, which is a model trained from scratch for the complex prediction problem with known stoichiometry. The main reference for this work is the bioRxiv preprint there by Evans et al. So there you'll see all of the technical details. But in the meantime, this slide should give you some idea of what we did. First, instead of training the model on individual chains, the training inputs now consist of whole bioassemblies from PDB. So like the little cartoon there, an example complex with A4B1 stoichiometry. And to help the model distinguish between chains, we added some extra input features, a bit like these numbers that are indicating the sequence and the copy number. Now, because a full bioassembly is usually too large to fit in memory, we have to crop out parts of it to train on. And this is done in two different ways. Half of the time we're choosing randomly chosen contiguous segments from the input sequences, but the rest of the time we're doing spatial cropping, which means that we pick an interface residue and include the other residues that are around it. Rationale for that is that the interfaces are really the thing you want a complexes model to learn about. So this is biasing the cropping towards interface regions to ensure that the model sees plenty of them. Then when building the MSA, we try and pair up sequences related to protein A and sequences related to protein B if they come from the same species. Those paired sequences go on the same row above the unpaired ones. And the intuition here is that it's helpful for the model to know not just that we have this grab bag of related sequences, but which ones actually coexisted in the same organism. And finally, because this complex has 4 chains with the same sequence, any permutation of those chains gives essentially the same structure. And so we take care to mark the model as correct, whichever version of the true structure it outputs. That ensures that it gets consistent feedback during training. So we evaluated AlphaFold-Multimer by running it on a large set of in-contact chain pairs extracted from recent PDB, filtered to remove anything highly similar to the training set. And what we found was an improvement over AlphaFold with this linker trick. It's a modest improvement for homomers, but quite a considerable one for heteromers. And so for heteromeric complexes in particular, we definitely recommend trying out AlphaFold-Multimer. And using a classification introduced in Capri, AF-Multimer produced an acceptable prediction in approximately 70% of cases and a high accuracy one in approximately 30% of cases. So we'd by no means describe complex prediction as solved. It's not as high accuracy as single chain yet, but this can already be an extremely useful tool for biologists. In terms of interpreting multimer predictions, most of the same advice applies. The key thing to remember, if you only remember one thing, is to use the PAE plot, not PLDDT, to judge the interchain confidence. So for this pentamer example, we've separated the residues corresponding to each chain using the red lines. And for the square that I've outlined in orange, which corresponds to the well-folded part of chain 1, you can see that we have high confidence in its position relative to the well-folded parts of all the other chains. Whereas for the residues outlined in magenta, which correspond to the unstructured tail of chain 1, we have lower confidence in its position relative to everything else, as you might expect. So as I said, this isn't yet a solved problem. So there are cases where we expect the model to do well, and there are some where we expect it to perform a bit worse. And this table summarizes some of the trends that we think we see. But that's not to say that the cases on the right aren't still worth trying. They will work some of the time. This is just a sort of statistical trend. Additionally, we still see false positives and false negatives. So if you find that A and B don't have a confident multimer prediction, it doesn't necessarily follow they don't interact in vivo. And conversely, if they do have a confident multimer prediction, then you can't be certain they would interact in vivo. But even with these caveats, we've heard a lot of positive feedback already from researchers about how they're using multimer predictions to help them piece together large complexes, or how it's suggesting new ideas for them to test experimentally. So that's incredibly exciting for our team, and we hope that there's a lot more of it to come. All right, so I'll finish on a really practical note by recapping how to access AlphaFold predictions and highlighting what's new there in the past year. So probably the easiest way to access a monomer prediction is via the AlphaFold Protein Structure Database that Samir mentioned. This is a website developed in partnership by DeepMind and EMBL-EBI. Comes with search functionality for finding your protein of interest, an interactive UI for viewing individual predictions, and you can download whole proteomes in bulk, which has proven very useful for bioinformatics applications. The biggest update since last webinar is that the database now covers almost all of SwissProt, as well as dozens of additional proteomes. So we're now up to about 1 million structures in total. So particularly for well-studied proteins, there's now a good chance that those are covered and ready to view both in our site and on Uniprot. We also provide feedback buttons now at the top of each page. So if you do see a structure you'd like to comment on, please do use those and tell us about it so that we can continue to improve the method. If you want to generate your own prediction, then the next simplest option is to use our Colab. And this is essentially a website that lets you run AlphaFold in the cloud rather than on your own hardware. The experience is kind of like using a prediction server. So you input your sequences, hit run, and after some time, the results will be displayed. And because you can edit the code before you actually run it, there is some scope to hack. So doing things like changing the inputs. And also community developed collabs like AlphaFold also exist, which provide a variety of other ways to run and to experiment with the AlphaFold model. The main update here is that our collab now supports Multimer. So if you want to try that out, especially for a smaller example, then collab would be a great place to start. And if you want to make predictions for very large proteins, on the other hand, or if you want to run a lot of things in bulk, then that is sort of less recommended in collab, just because we can't guarantee access to a particular spec of GPU or uninterrupted access over a longer period. And so for users interested in that sort of thing, for bulk prediction, we would still recommend checking out AlphaFold on GitHub. As well as the open source code, we provide a Docker image for easier setup and a simple launch script, which covers both AlphaFold and Multimer. The slight downside is that you do need some resources. Minimum is a machine with a GPU and enough storage space for the genetics databases. Although I believe we do provide a slightly smaller version of those now, which might suit some users better. Two things to mention here. Firstly, we have an issues page on GitHub that's checked semi-regularly. So if you have a technical question or a bug report, raising an issue is one good way to contact us. And you can also find out about new releases here by keeping an eye on our release notes. So some noteworthy changes in the past year have included, we changed our software license. So commercial use of AlphaFold is now permitted. This is where we posted the initial release of Multimer. And we've also updated the Multimer models recently, actually, in response to user feedback. So now Multimer is more likely to place a couple of chains far apart if it's uncertain about their relative position, as opposed to generating major clashes, which is making the predictions difficult for people to use. Okay, so it only remains for me to thank all the people who actually do this work and made it all possible. This is the combined author list of about 3 papers now, the AlphaFold paper, our human proteome work, and this Multimer preprint. We're also indebted to the much wider team at DeepMind, our collaborators at EMBL-EBI, and of course to the structural biology community who provided the data that AlphaFold's actually built upon. And with that, I guess I'll hand over to our next speaker, which I think is Randy Reed.
Okay, thank you, Kathryn. I think keep the questions coming and upload them so we can ask all the relevant questions at the end. And so let me just introduce Randy. So Randy works at the Cambridge Institute for Medical Research and is well known for his contributions to method development for structure determination using X-ray crystallography. And today he's going to talk about the impact of structure prediction on structural biology. Over to you, Randy.
Yeah, thanks, Samir. Okay, so Kathryn gave a great talk, and that's going to allow me to skip over a few things I was going to say, so that's good. Yeah, so I'm going to talk about the effect that AlphaFold has had on structural biology in practice. So before AlphaFold, like a little over a year ago, the big problem in crystallography was called the phase problem. So I've illustrated that in this little diagram here. It's a picture of 6 atoms. And if we measured diffraction data and we had no idea of what the phases for those structure factors are, we would get these blurry images like right now. But if we know the correct phases, then we get a very clear picture of what the structure looks like, 6 atoms here. So we can't measure this phase information directly, but if you know what the structure should look like and you can rotate it and translate it to put it in the right place in a model of the crystal, you can calculate the phases, and it's a really powerful shortcut. And it's gotten more and more important as the protein data bank grew, and now as we have AlphaFold models. So before AlphaFold, about 90% of structures were being solved by molecular replacement. They weren't all easy, and it was sometimes very challenging, especially if there were no closely related structures in the PDB, like nothing with 30 or 40% identity or better. So when you couldn't solve by molecular replacement, you would use experimental phasing, which is a method to perturb the diffraction pattern and measure data on 2 crystals or 2 different hands of the data, whatever. But it was significantly more complicated in practice to do. Okay, so why was molecular replacement difficult and what has AlphaFold changed? Well, AlphaFold and RosettaFold have changed a lot of things in this. The biggest problem was having a poor model. So if your model, even when placed in the right place, didn't put the atoms in the right place, then the phases you would calculate would be poor and you really couldn't solve the structure. This happened typically when you had low sequence identity, which isn't a problem with AlphaFold because the models hardly depend on the sequence identity of anything in the protein data bank. But what's still a problem is if the conformation is wrong. So if you have a protein that changes conformation and you only have a model of one conformation, you have to worry about that. Another issue that still survives to some extent is having a very incomplete model. So when you're rotating and translating a model of a protein, if that model that you're using is a very small fraction of what is in the crystal, the signal to noise will be very low. And it's much worse if you've got a poor model. So with good models, you can solve more difficult problems with more copies or smaller pieces, but it still can be difficult. What's more or less gone away is the case we often had where you had a complex or knew a protein and you knew part of the structure, but there was no protein of known structure for another domain, and that's pretty well gone away. Another thing that still exists is if you have poor data. So if you have low resolution, it's still hard, but it's even harder if you have a poor model. And if you have something funny about your crystal, some kind of pathology like these things I've listed on the bottom, that's also going to create issues. Okay, well, this is a chart showing the progress of all of the CASPs from the very beginning of CASP, I don't know, 28 years ago or so. At the beginning, so if you look at how difficult the target is on some strange scale that combines sequence identity and model completeness, it was very dependent on having a good model. As the years went by, until I got involved at about CASP 7, which is somewhere in here, things were improving, but it was still, the quality of models was very poor for the difficult cases. Then it started to improve in CASP 13 with the first version of AlphaFold, and then the second version of AlphaFold, which is the one we have now, where the quality of the model, so this is a scale of 0 to 100, where anything above about 90 is the kind of difference you'd expect to see between two structures of the same protein grown in different crystals. All across the whole range of difficulty, the models are all pretty good. But we have a little proviso we have to keep in mind. There are very few poor models, at least for isolated domains. So in CASP, models have traditionally been evaluated based on just the structures of the domains to give people some reward for getting things partly right. So as Kathryn showed, sometimes the relative domain orientation is not known, and that is something you have to keep in mind. Well, here's one example of the difference that AlphaFold makes now in practice. So I have in gray the experimental structure that was determined of a protein that was used as a target in CASP 14. And in sort of peach color is the closest homolog that existed in the PDB at the time, which is really distantly related, 7% identity, and that only covers half the structure. And this would be hopeless to try to solve by molecular replacement. With AlphaFold, the model of this protein is in blue on the right now, and they almost perfectly superimpose. And if you look at the details, it even extends to the details of the side chains in large part. So it's made something where you would have had to solve it by experimental phasing methods and turned it into something where it's an easy molecular replacement problem. Kathryn touched on this, and I think it's really worth emphasizing that having estimates of the errors in the models makes them far more useful than just having the model. So it's almost as valuable in some ways to know how accurate your prediction is as it is to have a decent prediction. So Kathryn explained these scores. So there's the LDDT score, the predicted LDDT score. It runs from 0 to 100 for rubbish to perfect. And As she said, anything less than 50 is probably intrinsically disordered. And there's also the PAE matrix, which tells you how certain the relative positions are of residues in the structure. Now we can use both of these. So let's look at this structure. I think it's the same one that Kathryn was showing us her beads on a string. It's the model of human fibronectin. All these domains strung together, and then these red bits of spaghetti or barbed wire, some people call it, which are probably completely unfolded. But the little domains of the fibronectin repeat domains are actually quite confidently modeled. However, if you look closely as they go by, you'll see that some of the colors are more towards the red end of the spectrum than the blue, telling you that they're probably not that confident in the predictions of those bits. So we know there from AlphaFold that there are better parts and worse parts and things we should just throw away. And one thing we can do is we can downweight the parts that are reasonably well predicted, but not as confidently as the best parts. And that improves the signal to noise. The PAE matrix in a case like this is the most important thing. So the PAE matrix tells us that we really ought to break this model up into a lot of different pieces. Now, if it's only a 2-domain protein, it doesn't matter that much because you could just look at it by eye and say, oh yeah, residue, whatever, I want to cut it out. And but in a case like this, it's quite inconvenient to do that. And Tristan Kroll in the group here has come up with a automated computational tool for doing this. So what he does is he looks at the PAE matrix with a community clustering algorithm that figures out which parts belong together. So which parts of the structure have dark green, basically, in the parts they share in that matrix. And by looking at that, you can cluster those parts of the matrix, and that breaks a structure down into a bunch of domains that are colored differently here, which really helps. And this has been put into some tools that are available in the Phoenix package through the CCTBX library, which is then used by CSP4. And I'm not sure if Chimera X is using the same code, but it has the same functionality as well. So it's very handy to break things up into the pieces that you expect to have different relative orientations. Okay, so there are, of course, some limitations in all of this. And the big thing is that AlphaFold was trained to recognize patterns from the structures in the PDB and the information in multiple sequence alignments. And if it knows anything about physics and chemistry, it's really only intrinsic. It's not explicit, it's implicit to what it finds in that pattern recognition. And that means anything that you need to know physics and chemistry to understand won't be reflected in the AlphaFold models. So for instance, it doesn't know about pH or ion binding or ligand binding. So because it doesn't know about ligand binding, it doesn't have a way of showing the effects of allostery when you have a compound that binds and changes the conformation of a protein. Another big one is everybody was excited because they would say, well, I'll just stick in my new sequence with a disease-associated mutation and see what the effect of that mutation would be. But the protein data bank doesn't have structures of proteins that have stopped folding because they've got disease-associated mutations and you can't model them. You need other tools. The AlphaFold models are a really good starting point for other computational tools that allow you to assess these things. And if you wanted a complete solution to the protein folding problem, it isn't here yet because AlphaFold doesn't tell us anything about the pathways by which proteins fold, or if you have structures that fold initially into some metastable state and then with some change in environment change to another more stable state, you won't get the different structures. In spite of these drawbacks, of course, it's already had a huge impact on how people do things in structural biology. So I've talked to any number of people now who say the first thing they do if they're looking at a new target is they look at the AlphaFold prediction, they look at the PLDDT and PAE matrix and design their construct based on that. So you know pretty well which bits to leave out so you don't have any unfolded protein that will mess up the behavior of your sample. And the PAE matrix gives you a suggestion about which domains interact strongly, so you know how you might chop the construct into pieces to look at different parts of the protein. then when you have an initial structure, if you compare it to the predicted structure might be distorted from the experimental structure, but it's usually some kind of smooth distortions with things locally being correct. That means, well, one thing that means is the sequence registration is usually right, so you don't have uncertainties about exactly how does the sequence line up in your tracing of the chain in the protein. But also, if you want to correct a model, then what you know is that a way to get there from and preserve what's right about the predictive model is to do something like morphing, where you just gradually, smoothly deform the structure. Then you can even go further. And this is, now I'm talking about some work that Tom Terwilliger has done in the Phoenix collaboration. So what he's been doing is iterative model building that benefits from AlphaFold in the middle of the whole process. So what you do is, after you've placed your model into the data, whether it's experimental, whether it's X-ray diffraction or cryo-EM data, if you see that it doesn't fit and you make some improvements to that model, and then you get a preliminary model, which might not be terribly accurate, but it goes in the right place, basically, you can put it back in AlphaFold as a template. You can provide your own template. And then generate a new AlphaFold model, and that usually follows the template a bit and ends up being better than either the initial AlphaFold model that you used to solve the structure, or whatever bit of model building that you did to make the preliminary model. And if you do this more than once, then you can converge on a structure that is a good model by the criteria of AlphaFold and also fits your data better. If you want to see something about this, Tom gave a talk about this, a webinar, and there's a YouTube video of that. And there's also a preprint of a paper describing this. What about on wider biology? Well, I imagine we're going to be hearing about these things in the second and third sessions, but basically it's like the whole genome revolution. Having a proteome scale structural database is enabling really new science. So one question that people have had for a long time is, how large is the universe of protein folds? Well, we've explored that universe much more widely with all of the AlphaFold models. It is predicting new folds, but is also establishing the limits of how many different folds there might be out there. Another exciting thing is if you do a structural comparison of all pairs of protein models in the AlphaFold database, you can find things that you wouldn't have suspected were actually related to each other, but they look like each other structurally, and that gives you some clues that there might be some unexpected evolutionary relationships. In terms of medical sciences, I think the thing here is to think of the AlphaFold models as a hypothesis, and you can take a hypothesis and generate other testable hypotheses from them. So here's a kind of important example. This is a protein that took us several years to solve. It's the structure of iduronate sulfatase. And if you have mutations in this protein in the wrong place, you have something nasty called Hunter syndrome. So on the structure, I've colored in magenta all of the residues that have been mutated in cases of disease that have been discovered. And by mapping these on the structure, we could then come up with a reasonable hypothesis for why each mutation causes the disease. Usually it's not by affecting the enzymatic mechanism, but rather by just making the protein less stable. Well, in most cases, I think an AlphaFold model will be good enough to generate the same kind of hypotheses, which you could then test by making site-directed mutants and measuring protein stability or protein activity. You can also figure out which residues might be involved in an interaction. That allows you to design a site-directed mutagenesis experiment to see if that's true. You can look at the structure and see, are there any clefts that look like drugs would bind in useful places? So it can help you to decide whether a potential target might be druggable. And Kathryn talked about this, but people have been looking at looking at whole proteomes and trying to decide which pairs of proteins interact and how do they interact. Okay, so what's going to be in the future? Well, there will be some things to improve monomer models. And one thing that Tristan pointed out to us a while back is that if you look in the fine print, it turns out that the AlphaFold training data included every structure in the protein data bank with resolution better than 8 angstroms, which is like incredibly low. And the data that's used could be quality filtered, and that might have an effect on removing decoy conformations, like conformations that you don't see in good quality proteins. But more importantly, I think the multimer models are not, as Kathryn said, as good as the monomer models. And there's a lot of room, I think, for improvement in how you make paired multiple sequence alignments. So it's really a very difficult problem because it's hard to know what's the difference between a homolog in two species where there is a homologous partner or a paralog which doesn't actually bind to a protein of the same type. So very difficult to do the multiple sequence alignments in a way that pairs things correctly, and there's intrinsically less evolutionary data to look at. Could we get around the problem of the model not fitting the actual experimental data or not obeying physics? Well, potentially you could imagine doing that by adding more tracks to the data. So instead of the multiple sequence alignments and the pair interactions, you could also add later on in the process, when you have a decent model, you could add a data track which says how well does the model fit the diffraction intensities or the map, or a physics track which says how could we improve this model to make it satisfy what we know about physics and chemistry. Overall, I think it's had a big impact already, but it's going to continue to have great impact on what we do, and lots of clever people are going to do a lot of things next. I just wanted to end with a few acknowledgements here. On the top left is the people in my group who have contributed to the kind of analyses that I've talked about. On the right are people in the Phoenix project, Tom Terwilliger and Jane Richardson, that we've had some nice chats to. And the Wellcome Trust, which has funded most of our research over the last couple of decades. Okay, thanks for your attention.
Thank you very much, Randy. So we go to the next presentation from Sergey, who is a John Harvard Distinguished Science Fellow at Harvard and has been actively working with AlphaFold and RosettaFold over the last year. So just to mention two of his papers, the eukaryotic complexes that David Baker's team published, he is author on that, and also a couple of papers on de novo protein design, just to mention two. And he and his colleagues have also developed CollabFold, which I'm sure many of you know about or have used. And today he's going to talk about impact of structure prediction in AlphaFold, RosettaFold on structural bioinformatics. Sergey?
Oh, thank you, Samir and AJ for inviting me. Very excited to be here. So I'll talk about structural bioinformatics and how it sort of changed a lot of things in the last year in terms of being able to sort of classify proteins, trying to understand where proteins of unknown function, like what they might be doing. So I've been sort of working in this area, and also other people have been working in this area. And one thing I really wanted to do is, let me set a timer for myself. Is to give you some of the problems that we saw and things that maybe you should be also watching out for when you're doing similar analyses. So the roadmap I'll follow is give a quick introduction to structural search. You can think of it as like BLAST, but with structure. What are these structural searches good for? Some tools, and I'll describe one tool from a colleague of mine, FoldSeek, pitfalls to watch out for, and some recent large-scale efforts to try to classify AlphaFold-DB. I'll just show like a slide on that because there's been some preprints coming out now recently about this. And also, if we have time, I want to touch upon some interesting applications of AlphaFold for hallucination of proteins. And even though we think of this as engineering, I think there might be some interesting applications towards bioinformatics, where you can think about like hallucinating the unknown space, and then maybe linking up some folds and so on. I guess the big picture here is often when we have a single sequence, we might not know what it does. And so if we look at the sequence and we compare it, search it against a database of other sequences, we can construct a multiple sequence alignment. Now, if one of these other sequences from these other organisms actually have a known function, then you can say, well, this looks really similar to this. Maybe my human sequence also does that same thing. But sometimes it doesn't appear to have any similarity to sequences that are close by. So one thing that's traditionally been done is to convert these sequences to a profile, which is just a probability at every single position. And then once you have this profile, you can search against a database of profiles. And by the way, I'm including like different software names here just that do this kind of work. And then maybe this profile looks like an existing profile somewhere in a database of profiles. And so in this case, it kind of looks like a transporter B. But sometimes it has no hits. And so then the question is, what do you do when you have no hits? And one thing that's been really exciting is when you do have a multiple sequence alignment of related sequences, you can look for these patterns of coevolution. And so, for example, this position appear to be covariant with each other. And often that indicates that these positions might be close together in 3D structures. And so we think methods like AlphaFold and RosettaFold sort of exploit this information from multiple sequence alignment to predict the structure. But once you have a structure, instead of doing like a sequence or profile search, you can do a structural search. In this case, like, oh, the structure looks a lot like this other structure. Maybe it actually does something very similar. But then the question is, what is it good for? Well, one thing that's really good for, and this is an example that I like to use, and I've actually now looked to see if AlphaFold made a similar prediction, is this is a protein from Bacillus that no one really knows the function of, no one really knows what it does. It still labels an unknown function. But if you look at the structure that's predicted by us and then also by AlphaFold, it looks very similar to aquaporin. In fact, some of the same amino acids at the interface here between these two helices look very similar. And so just based on the structural similarity and then also based on some key residues, we might think like, oh, maybe this is also an aquaporin. This is a really interesting example because it's actually in this case, the folds are actually quite different. The connectivity is quite different, but the architectures are almost the same. And so these are kind of the insights that you might be able to provide based on this. But you have to be careful because just because two structures look very, very similar doesn't necessarily mean that they have the same function. And one thing that we like to do to sort of get around this problem is to look at conservation. So any predicted structure, what you could do, assuming you have access to the multiple sequence alignment, is to map the conserved residues onto those structures. And so once you have the conserved residues mapped onto the structures, then once you do have some kind of similarity, so in this case is an example by Nick, Christian, and Lisa, is in this case, this protein look very similar in terms of fold, but if you also look at where are the conserved residues, they have the same identities. In this case, I don't remember which color means which amino acid, but in this case, we'd say, okay, maybe these guys are actually doing the same thing. So just because they have similar structure does not mean that they have the same function, but if they have the same conserved residues in the same locations, that gives you more evidence towards that. The other thing, and this is something I personally work on, is saying, well, can we find some really distant similarities? So one hypothesis is that maybe there are sort of symbiotic communities in the ocean somewhere that resemble some prehistoric times where you have organisms interacting with each other, and that was sort of the origins of the eukaryotic cell. And then maybe some of the proteins that are involved in interactions between cells, for example, this community are the same proteins that are involved in cell-cell interactions in, say, a multicellular organism. And if we could find sort of these similar proteins by structure, maybe we can make some of those connections. And so this is an example of two proteins that share less than 10% identity, yet structurally they look almost the same. But that being said, you have to be careful because just because the structures look the same doesn't mean homology. I mean, these two things could have just evolved completely independently in a convergent evolution. And so one way to potentially argue that maybe there's some homology is to maybe look for some missing links. And I think people sort of done this with sequences. Say, okay, if you get one sequence and then you get another sequence in there. But one thing that I think was really exciting with ability to have models from AlphaFold is that maybe you could do some kind of structural linking. And so let's say if this protein have very low probability that they're the same from, let's say, HE search, but maybe there's an intermediate structure that has a high probability, and then you can link them up. So this is an example from Nick Christian using some crystal structures. But we think maybe this could now be done sort of at scale potentially with models. And so you say, okay, I can sort of maybe link up proteins that way. So you have to be careful not to argue that it's homology just because of similarity, but maybe there's some ways to potentially add some evidence towards that. So now, how do you actually do this? One cool method that's been available online now for a bit is fold-seq. And fold-seq is you could literally just throw your structure prediction, your AlphaFold prediction, or maybe your crystal structure in here, and it will identify hits for you. And this automatically searches against protein databases and also, PDB databases, but also AlphaFold predicted models. And it works literally in seconds, which is quite exciting. So if you're interested, go to foldseq.com. And the way this actually works, the reason why it's so fast, and these are some slides that I stolen from Martin, is that it takes the structure and sort of breaks it into effectively a sequence. And so the way that actually works is you could take any given pair of residues. So for example, let's say I take this blue residue and I identify its closest neighbor. And based on this neighboring information, I can extract these vectors that describe the angles and dihedrals and so on, and then encode them with a variational autoencoder and then convert them into some kind of alphabet. And once you have effectively a sequence that tells you for each position its neighboring and its environment, you can then align these sequences as they were actually amino acid sequences. And so it works almost as fast as doing sequence search, because for every position you're encoding its environment. But you have to be careful though. So because the question is like, okay, if you do a search, does it mean that it's actually no hits? Is it like a new fold? And so one thing, as Kathryn pointed out earlier, is you have to be really careful about how you pick out your models. So let's say you have this prediction and it has a really, really high PLDT value, like this is colored by PLDT, but in fact, there's two domains, and this domain is really confident, this domain is really confident, but the interaction between the domains is not confident. So if you search for this whole protein and you found no hits, it doesn't mean that there are no hits. It just means that this particular arrangement of these domains has no hits. But as you could see in this PAE plot, there is no actually evidence that this arrangement is correct. So these are sort of arbitrarily placed. So in this case, it makes sense to sort of chop this, and then search for this and this individually. Furthermore, you might want to remove like maybe long loops that are maybe disordered based on the predicted PLDT values. The other thing to watch out for is a lot of times these programs like fold-seq, they provide a TM score or some value scaled by the prediction. So this is the query that you provided, but you might also want to look at the scaling based on the aligned residues. So for example, this appears to not match this, but if you look at the other direction, it actually has a pretty strong hit. So you have to look at these partial hits. And finally, the other thing to watch out for is a lot of databases, they sometimes provide all proteins as things that are chopped into domains. And so imagine if I had searched this against this and this was sort of just chopped in the middle, it might appear it doesn't match it. But if you search against full chains, then you might actually match it. So things to look out for. If you don't have hits, doesn't mean there's no hits. It's just that you might have to do some processing to your models before you're doing that search. The other thing that I like to point out too is like, even if you think you found a hit, like let's say somebody gives you a hit. And so one common software is TM-Align. And often people like to say, oh, 0.5 TM score means the same fold. But this is an example where this is one protein that was designed, another protein that was designed, and by TM score, they have a really high TM score, 0.6. And so they say, okay, this is the same fold. But if you actually look at these, they're actually quite different in terms of, even though their secondary structures align up and their architecture is the same, their fold, I would argue, different, because this one actually forms a knotted protein, while this does not form a knot. But you'd still get a pretty high TM score because there's a lot of partial overlap. And so even if you have a really high score, it's still worth loading up the models and looking at them and say, okay, is the connectivity between secondary structures correct? So you can't just blindly trust any numbers that come out. You should really look. And so one thing that's really cool about fold-seq is it will give you back the alignment, and you can really look at it and say, okay, do I really trust that alignment? Besides fold-seq and this alignment really fast with sequences, fold-seq actually does provide ability to do TM-aligned search as well. But one thing to be careful about is it does do some pre-filtering based on this sequence. So if it finds nothing, maybe it's possible it got removed during the pre-filtering. A few other software that now implement AlphaFold database within the web server include DALI. It's very slow, but it's considered would be one of the most sensitive software. So if you don't find anything with FoldSeq, don't give up, try Dolly. But also these other methods, which I haven't tried, but implement AlphaFold. And so I think it's also worth checking these out if you don't find any hits. This one's supposed to be also a very fast alternative because it encodes the sequence as characters based on just the local torsion angles. And also from Kahira's group, also worth checking out. There's been recent efforts on classifying structures from AlphaFold, including Kath. Christine talked a lot about this kind of stuff, and they found quite a few new folds, which I think are quite exciting. Here's a bioarchive link. Also, Nick Grisham has been working, and his team has been working really hard on classifying all the AlphaFold structures. And they're not yet publicly available in the database, but it's coming soon. So to keep eye out. But in both teams, both Christine and Nick, they have both reported that they have seen things that they believe to be new folds or new superfamilies that were previously not in the PDB, which is quite exciting. So like something like this looks kind of something you probably haven't seen it in a while or in the PDB yet. Now, just to switch gears a little bit, and this is more of a section just for fun, protein hallucination. So once you have a model that can predict stuff, can you just invert this model or just start to hallucinate new protein structures or sequences? And the general idea is, imagine you train a model that takes pictures of cats or dogs or fish and classifies them as such. And so if you take this sequence and you classify it as such, later on what you could do is you can give this model complete random noise, and as probably would predict, the model say, okay, I don't know what this is. But let's say it has like a slight ink towards cat. You can say, hey, maximize the probability of cat. And if you just keep iterating, like keep changing this input until the classification becomes a cat, often what you'd see is an image of a cat appearing in this noise. And so similar ideas could actually be used for these models that were designed to predict protein structures. So just to give you an example, just another one interesting example is here. Imagine you're looking at a bunch of clouds and you stare at these clouds and you're like, okay, I think I see a fish in there somewhere. And so then you could tell the model, okay, keep changing this picture until the probability of the fish is really, really high. And if you just keep doing that, what people have noticed is that you can actually convert pictures of clouds into actually really cool artwork. And so the idea is that you actually use these very similar procedures to methods that were designed to predict protein structures. So just to give you an example, so methods like TR Rosetta, you can give it a sequence and it'll predict back a sort of a distance matrix for every pair of residues. And initially the prediction is very, very noisy. It doesn't know what's going on. It's like, I don't really know if it's a helix, it's a sheet, what's going on here. And so then you say, well, whatever you see, just enhance, just make it as high probability as possible. And if you do that for actually many 10,000s of steps, eventually you get back a very, very clear image, and that folds up into a structure. And there's been some now experimental evidence showing that these things actually do fold up into structures. On my side, I've been actually thinking, can we actually do the same thing with AlphaFold? And I guess the answer is yes. Computationally, yes, we can. Like you could take a sequence, give it to AlphaFold. Initially, it's not confident about it. And then you just keep changing that sequence until it becomes really, really confident. And we do implement this package inside Colab. Design is what a new thing I'm working on. But that being said, you have to be careful because these sequences could be adversarial. So it could be because you can come up with sequences that trick AlphaFold. But we still think these are kind of interesting because it allows you to generate backbones. Then these backbones could be used in other applications, then be redesigned with other cool software. So it could be used to generate some initial hypotheses, which then could be refined. So I wouldn't say use these as a truth, but could be really interesting to explore. One really cool application that just got released is Basil and collaborators have used AlphaFold to generate a whole bunch of different symmetric homoligomers. And here they make a space of all the PDB homoligomers and their designed homoligomers or hallucinated homoligomers. And they say, hey, we found some things that look like nature has never ever done before. And one thing they do find is that you take these sequences directly from AlphaFold and try to express them, they actually don't work. But if you redesign them with additional software like various MP&N software, you can then actually get these things to work. And they've experimentally now confirmed them. So AlphaFold still is a useful tool as a generator of structures, but just be careful not to use it But the reason why I bring this up is because I kind of thought it was funny, because as soon as I saw Basile's post, I started looking at these structures that he posted. And then I remembered a post from Christine's group where they showed some structures. They say, hey, these are new folds that were never encountered in the Cath or PDB. And so if you actually look at them really, really carefully, it kind of looks like there might be some similarity there. Like you still have like 3 sheets and a helix, 3 sheets and a helix. I guess to be fair, it actually looks quite different. I mean, there's 5 copies, while here there's 3 copies. But it did make me wonder, like, oh, could this be a way to potentially fill in sort of the missing links of protein space and maybe link up some proteins that we've sort of been comparing to each other before, but never really had those alternate paths. So maybe we could sort of fill in a lot of things, and that could be kind of fun to do. Besides us, there's been actually quite a lot of people trying to use AlphaFold for sequence and structure generation. Some of them have been experimentally looked at. But I think there might be some interesting applications of bioinformatics towards this. Is like if you can sort of find some missing links between folds and so on. Well, I think I might have finished a little early, but I'd like to thank various people on the Collab Fold team, FoldSeek, ECOD for providing some slides, and also Kath folks. Thank you very much.
Thank you very much, Sergey. Now, the first question to Kathryn. You said about recycling, and how does increasing number of recycling steps affects the outcome?
Sure. So if it's already a protein that has a confident prediction with the standard number of recyclings, we wouldn't expect any change. However, we do observe rare cases where you will get a more confident model by running more recyclings. And so I think we only saw that once in CASP, which is why I say it's rare. That was with, I think, T1064, which was a SARS-CoV-2 protein. And that one was challenging for the model to predict. And so if you ran more recyclings, we ended up getting a better prediction there. Something that I would like to see before making that part of a standard workflow would be what it does for, say, unstructured regions or something like that. I just want to sort of check whether the model will hallucinate a structure if you recycle for long enough before making that standard. But it's a thing that is available to try, certainly in some collabs.
Okay. Then there is a general theme over what's coming next and what it can and cannot do. So let's take all of that one by one. So first question that came in before the webinar started. So just to remind everyone, you can send in questions before the webinar, is about dynamics. And is there a plan to think about how AlphaFold might also provide information on protein dynamics?
As in a DeepMind plan, or is this a general question?
So you can answer from DeepMind point of view or general, and Sergey can add. I'm sure he has done a bit of work as well, and Randy might have seen some of these things as well.
Okay, so I would say nothing, that we're going to put out imminently in terms of tooling or something like that. From our perspective, I would see that as being a huge new problem because, as Randy was saying, this is really a model that doesn't, explicitly know anything yet about protein physics, at least from my perspective. And so, I would see that as a very long-term research project personally. Definitely something that would be interesting to work on, but not something that we're going to, publish next week.
Okay. Sergey, you want to add anything on dynamics?
Oh, yeah. At least in our experience, I mean, what you could do is sort of enable the stochastic part of AlphaFold, and sometimes you can sample from the uncertainty of the model. But often if you have a really good MSA, even if you perturb that and do dropouts, it doesn't change anything. There have been some hacks where somebody purposely took an MSA and reduced it down to just a few sequences, and that seemed to help make the model less certain. But whether or not that could actually be used to predict real conformations, or if that's just, you could just sort of sample in between, it's not really clear yet. But that's the only hacks that I've seen so far. Like you purposely make the MSA small, that you disrupt a lot of the coevolution maybe, and that sort of allows you to maybe jump between different structures.
Yeah, I think the important thing is you can always generate many structures, whether that means you are seeing dynamics is a completely different thing.
Yeah, I would like to see the physics added to it, but I think it's not necessarily trivial, because in CAST 14, there was a refinement task where people were supposed to take the best predicted models and refine them. And a lot of them ran molecular dynamics with some constraints. And basically the AlphaFold models were unimprovable by those methods. So I think you'd need to combine different things together to make it all work.
Okay. And then there are questions about complexes, and that includes protein nucleic acid complexes, so RNA, DNA, protein ligand complexes. So where the field is in terms of complexes, and what are the next things that we will see coming up?
Okay, so at least in terms of where the field is, I think I may have already said, as much as I can on that topic in the slides. I think that we are in a very exciting place for complexes. I don't want to be too negative on that. I'm a special fan of all of the work where people are taking cryo-EM maps and using Multimer predictions and AlphaFold predictions to sort of fill in the molecular details of those. I also do think that our current complex prediction tools have some potential for interaction prediction. I just don't want anyone to get carried away and think that, because they don't observe a confident AlphaFold-Multimer prediction, that means that A and B absolutely can't interact. I'd hate to send someone down the wrong path because of that. But we're already seeing great applications in those areas. I think, there's room for improvement, certainly, and that if we could get it up to the same standard as single chain prediction, then it would be much, much easier for people to, have confidence, for instance, when they see a negative, that it's actually non-interacting. That would be wonderful, so that we could build out networks. Better interface prediction would make it much easier to build out complexes, so we'd love to see that. In terms of things like nucleic acids, the thing that worries us there, I think, is probably the lack of data. There are just far fewer PDB structures, at least for RNA, I think, and also for DNA. So perhaps if somebody came up with, a great new assay or a new data set, that would make it a more appealing ML problem there.
Okay. Randy, Sergey, you want to add anything to that? No? Okay.
I've seen one effort where people sort of adapted a similar architecture to RNA structure prediction using a multiple sequence alignment, MSA transfer, I think from Zhang's group.
Yes.
That was the only effort I've seen so far.
Okay. Then some questions about the AlphaFold pipeline. So the minimization step, is it using force fields or QM?
Yeah, that's a force field step. So it's using OpenMM with an AMBER force field.
Okay. And are there any plans in terms of thinking about post-translational modifications or ligands?
So not plans from me. On PTMs, I think the thing that I would be most curious to understand is what people really want from models like that. So would it be valuable for us to put out a model that is, predicting exactly where these modifications sit structurally, or is it that people want to know which residues are modified, or do they want to know by which protein? I'd be really interested in having some conversations with people about that, just because for us, it's important to pin down exactly what's useful before we start working on an ML problem like that. So that's where I stand on PTMs.
Okay. And there is another question about, and you mentioned it in your presentation as well, antibody-antigen complexes. So do you want to comment on whether that is something that DeepMind is interested in? So there are other papers that have come out, but that will come to that. But to begin with, do you want to comment on that?
So personally, I see that as fitting into the sort of more general category of small MSA or MSA-free. I don't know if everyone on the team agrees with this assessment, but personally, I'm an antibody pessimist. I think that it is going to be a much harder problem because of the lack of evolutionary information. And so if I were to work on a problem like that, I think I would probably come at it from the perspective of trying to reduce the MSA size, trying to develop general models that are, able to cope with MSA-free. Just because then you have so many more test cases and much more data as well to approach the problem.
Okay.
Samir, getting back to the post-translational modification thing, there is an initiative called AlphaFill, which is where you take things that you've seen in the protein data bank that are similar, and then you just transplant the carbohydrates or the ligands into the structures.
Yeah.
And that can be quite effective.
Yeah. Sergey, you want to add anything on the antibody complexes and prediction of those?
I haven't looked at it too much, but one thing I do wonder sometimes is one thing we see is during the recycling of complexes, what you see often is the monomers are almost correctly predicted right away, and then they don't change at all. While I'm wondering if for antibodies, antigens, if there's some kind of confirmation that happens upon binding. So I think one hypothesis is maybe what's happening is that AlphaFold identifies maybe some surface patches and then brings them close together. But if the monomers are mispredicted to begin with, the loops that maybe bind, if that could be somehow. So I think maybe some kind of confirmation of change upon docking prediction would be an interesting way to maybe solve that problem.
Okay. And then there was a question that was submitted before the webinar about usefulness of these predicted models in drug design, and what the limitations are and how much can we actually do with the predicted models. Who wants to start? I'm sure all three of you have comments on this. Randy,
Please, someone else go.
Randy, you want to start?
All right. I think they're probably similar in their utility to the initial crystal structure that you get of something, but you need the feedback of seeing how things bind and you know, the iterative nature of it. So you aren't going to get away right now from the structural biology aspect.
Okay.
Yeah, I mean, even with a crystal structure, drug design is still hard, right? There are so many more stumbling blocks that are to do with biological complexity as opposed to just the structural biology of the thing. I think in our paper, we sort of compared it to, you know, having an unliganded structure for the first time. So, you know, in terms of docking, you're going to find that perhaps some side chains are not in the right conformation to receive a ligand or something like that. So that might be a useful comparison.
Okay. Sergey, you want to add anything to that? I mean, there are, again, many more papers on this, and a lot of work being done. So it's very difficult to cover all of that.
Yeah, I'm honestly embarrassed at how many you know, interesting papers I'm forgetting to mention here. So thank you for everyone else chiming in.
So there is another point about AlphaFold and probably Rosetta Fold as well. So in PDB, we see multiple conformations of a certain protein. And then if you try and predict maybe that structure, AlphaFold sometimes or predicts only one of them. Why is that? And is that something that you are looking at? And can we kind of use this information?
I mean, I feel like Sergey has probably already come close to answering that with his comments about sampling from uncertainty. If I wanted to try and sort of trick the model into generating more diverse conformations, one of the things I might try is exactly that, just reducing the size of the MSA, because sometimes, you know, with a very deep MSA, AlphaFold is so confident in one particular conformation, that's all it returns. So I would try running more models and probably reducing the MSA size just to see if it gives something different.
Okay.
Yes, one thing to add to that is the one thing that we see is we believe that AlphaFold uses the convolution information, and so exactly like we were saying earlier, if the contacts are really strong, it will always go for the structure that has the most contacts. And so one hack you could potentially do is if you know which residues are at that interface of one conformation, if you take the MSA and just literally X out all those positions across all those columns, you will actually get the other conformation. Because what that does is effectively kills some of the contacts that are from co-evolving, and then the other contact can now be satisfied. So you see like two potential ways, like you purposely reduce the MSA and you kind of hope that it kills some of those contacts, or you could, if you somehow know which might be the interface that you want, you can figure out, okay, these are the positions and I'm just going to kill them all and then maybe I'll get the other structure.
So without knowing that, I remember Debbie Marks had some work a number of years ago talking about being able to parse these things in terms of mutually exclusive sets of contacts. And if you could do that, then you could favor one time and favor the other another time.
This is the EV couplings. And also, Randy, you mentioned a case where providing AlphaFold with a template, which is modified based on experimental data, does produce a different structure. So that's another kind of example. And then we come to the intrinsic disordered proteins, and there are a couple of questions related to that. First of all, how does AlphaFold perform on intrinsically disordered proteins, and what have you observed? Let's start with that.
Okay, well, that's kind of a difficult question, because how it performs is, it's a structure prediction algorithm. And so it's not going to be able to predict a structure for something that's intrinsically disordered. We do think that it's reasonably good at picking up on those regions and assigning them low PLDVT. And that's something that other people like Balint at EMBL and others have also, independently confirmed. So I think it will help you identify them. It won't necessarily tell you much beyond that. I think I saw a question about relationship to disease as well. So for something like misfolded proteins, often that's very dependent on the sort of environmental conditions the protein finds itself in. It can form different kinds of aggregates, even, depending on that. And these are factors that AlphaFold doesn't know anything about. So beyond saying that it's intrinsically disordered, I don't think it will necessarily tell you anything further.
Ask a follow-up question, though. So if you have an intrinsically disordered protein, sometimes the part of it becomes ordered in the right context when it has the right binding partner. So do you find that if you use AlphaFold-Multimer with the right binding partner, that you get the right confirmation for some of these?
Sometimes, yes. That I have slightly more hope about. It does rather depend on you knowing what the right binding partner is, of course. Yeah. And I wouldn't say that, it's something that I've observed frequently. But if I wanted to know more about an intrinsically disordered region and thought that I knew the binding partner, I would absolutely try running it through AlphaFold-Multimer. It's not a complicated thing to try. So yeah, I'd take a look and see if it generates a useful hypothesis.
And just another thing to add to that, even with AlphaFold, I think Norman Davey and Balint, they have shown that some motifs which are in the disordered region get a secondary structure, and those are in some cases they have found regions which actually bind when the complex is made, or in the right context. So sometimes we see in the AlphaFold structures a big disordered region and a small helix in middle of that, and some of those are kind of relevant confirmations, but it's very difficult without the context to say anything more on that. But again, there are more than one paper, I think, related to this. So another related to what Randy was saying about adding physics. Does AlphaFold perform protonation with physiological pH and ionic strength before energy minimization? And if not, will doing this, you think, will improve the accuracy?
So I'd be doubtful that it would improve the accuracy. No, I don't think we do that. I don't think the model knows anything about these sorts of contexts. So it's an interesting point, but yeah, I don't think that's something we're looking at.
Okay. And then a practical question. If we want to use AlphaFold and the sequence is longer than 1500 amino acids, what is the recommended technique to fragment proteins greater than 1500 amino acids?
Okay, so I can say what we did in the database where we had to automate the whole thing, which is, in that case, we just tried to keep it under, about 1400 residues, something that's not, basically try to pick a length that's not too inconvenient for you to fold with whatever setup you're using, and try not to go above about 2000 residues, that would seem reasonable, and also include some overlap for your initial attempt. Just so that you don't get any domain that's being bisected because it's right on the join between two segments. That would be kind of annoying. And then if you're not doing this for some sort of bulk application where you don't have the opportunity to refine, You can always come back and look at the PAE afterwards, and then it should be reasonably easy to identify domain boundaries and fold individual domains if that's what you want.
Okay.
That would be my suggestion.
Okay. How does AlphaFold compare to Rosetta Fold is a question everybody asks. So Sergey and Kathryn, what do you know about that?
From personal experience, not that much, because I haven't really played with Rosetta Fold. I do know that in the most recent paper of theirs, it wasn't quite matching the same accuracy on the CASP set. So that is as much as I know on that.
Okay. Sergey, you want to add anything?
Yes, actually, you know what? Maybe I'll pull up a slide really quick. Actually, Randy, if you have any experience using one or the other, maybe you can speak a little bit while I look for this.
Well, I ended up contributing to the Rosetta Fold paper, looking at molecular replacement models generated by Rosetta Fold. And basically, when AlphaFold became available, those models were a bit better in general. I think the Baker group is making progress on making a newer, better version of Rosetta Fold. So who knows where it'll be in the next iteration.
Yes, so I think the original published version, as Kathryn mentioned, it was not doing as great. But Ming-Young from the group has been making quite a lot of progress. Also, Frank DeMaio and Hustis. And so I think at this point we have caught up. By we, I mean mostly Ming-Young. And I don't know, like a few months will we actually get better or not. But and so this version is currently the public version. This the version is currently being tested during CASP. And so after the CASP experiment, this will also be released as part of the Rebetta server and also into a Google Colab notebook, hopefully by me later. I guess the current public version, to answer your question, is not doing as well. But also one thing I should also mention here is that even though they're very similar on average, it turns out that sometimes Rosetta Fold gets it right, sometimes AlphaFold gets it right. So there might be scenarios where if one method doesn't do well, it's almost like somebody made like the sixth model, I guess you could think of it. If we think of AlphaFold has 5 models, and also as other people are training it, you have additional models to run, I think would be quite interesting. Okay.
So following on from that, is there a difference in performance on membrane proteins versus water-soluble ones?
So you can definitely generate good predictions for membrane proteins. There were some of those in CASP, for instance, and we performed reasonably well on those targets. I think one thing I would point out is that sometimes it distorts long helices. So you can end up with situations where 2 ends of a protein in a single pass membrane protein end up close to each other, which of course is unphysical. And the way that you can tell that that's happened is, again, by looking at the PAE plot. So you'll see that it doesn't actually have confidence that these 2 domains are close to each other. It's just, the helix has become distorted. So that can be an issue for membrane proteins. And one thing that I would recommend for trying to sort of fix those examples would be, if you know, the stoichiometry, the binding partners, try a multimer prediction for those, and you might be able to get something more informative.
Okay. Anything, Sergey, you want to add based on your experience with AlphaFold?
I think the same thing as Kathryn was saying is that sometimes you do see things that are from 2 sides of the membrane. Like it doesn't know that there's a membrane there, so it's a so you do sometimes see those artifacts.
Okay, so then you mentioned multimer. So will prediction of multi-domain proteins or also the membrane proteins, etc., be better from multimer rather than AlphaFold itself?
So I wouldn't necessarily expect it to make that much difference for a single chain protein where you're looking at domain packing, because I think part of the extra complexity with multimers is maybe in the MSA and in the pairing. I don't know for certain, but I would imagine that that's part of the difficulty. And also, the trading setup includes complexes for multimer. So those are, 2 big changes, and I don't think they would necessarily benefit in terms of domain packing. But I will admit it's not a thing that I've actually tried. So whenever someone asks something like this, I would always encourage them to run even a couple of dozen examples in Colab and just try it out, because then you'll get an empirical answer instead of my opinions and views on these things.
Randy, any experience on the experimental structure determination, domain orientations, etc.? How do you handle that, or do you only use individual domains?
If the PAE matrix says that we shouldn't trust the relative orientation, we cut them into separate pieces and place them independently. Works much better.
Okay. A question about the AlphaFold database, Kathryn. Is AlphaFold going to update models deposited in the database? And all models, at least for human, seem to be there from the beginning, but are they going to be updated if we get a new version, better version?
Yeah, I think if we ever, develop a significantly better version of AlphaFold, then of course it would make sense to update. We do, want to be conscious of the fact that we're already asking our EMBL collaborators to do quite a lot in terms of the database.
Yeah.
So, recently the focus has been on expansion, like getting all of SwissProt covered as a priority, and then expanding to a larger proportion of, known sequences. But once we have the coverage up, I think looking at updates and versioning will be one of our priorities.
Okay. Can you say a little bit about the training set that you used? Were there any specific choices made? And are there ways in which this can be updated rapidly with new training sets?
So as Randy mentioned, we're not actually very selective in terms of the things that we include in our training set. So for instance, including things with very low resolution. I think we tend to, come at it from the perspective of more data is usually better there, for the initial training. And small changes in the training data often come out in the wash, so you'll find that it doesn't make a huge impact on accuracy to restrict that. In terms of whether we would update the training set, we've actually found it really convenient to not update. We've been using the same training set since CASP 13, I think. And a nice thing about that is that it gives us more examples that we can test on that the model hasn't seen before. So if you want to know how AlphaFold does on a particularly biologically relevant class of protein and structures don't come into the PDB very often, it's nice to have 2 or 3 years worth of PDB that the model hasn't seen, just so that we can, look at that. And so internally, at least, I don't see us updating soon.
Okay.
So I had the impression from something that I read that at least part of the training data was updated in January or February of 2021. So did I misread something, or is there some other aspect of this?
Yeah, so that's not the training data, that's the templates that we allow the model to have access to, because we can still search a different template database, even if it's not something that the model's, been trained on.
Right, so I can go to somewhere, I can go to CollabFold instead of the AlphaFold database, and I can get something uncontaminated by that newer information.
Yes, for instance, you can use a newer version of something like PDB 70 or whichever template database is underlying that.
Yeah, thanks.
Okay. And can you, we don't seem to have much time left. Maybe what we can do, I think, is to save some of these questions and send it to you, and you could try and answer as many as you can, and we can put them back on the web pages. Some of these questions which are at the top, I didn't ask because these were more generic questions about nucleic acid complexes, etc., and the oligomeric states and prediction of oligomeric states, etc. So I think we will save these questions and then send it to Randy, Kathryn, and Sergey and get their answers and make those available as well. So thank you again, Kathryn, Randy, and Sergey for joining and giving excellent presentations and answering all the questions. And thank you all who joined, and see you at the next webinar. So please register, please send in your questions. And hopefully we already have more than 500-600 registered participants. So it's going to be another exciting webinar like this one. So see you next week. Thank you very much.